All right. Hi, everyone. Thank you so much for joining our September PyData Meetup. I can't believe it's already September. It's so crazy. But we're really excited to have you all here today. And we're very excited to have Xavier-Lewis Palmer be our speaker. And he is a full stack biotechnologist with a PhD. And the topic for this month is machine learning in biomedical microscopy. And before we begin, we're just going to hop through a few announcements. We do have some open roles available on MetroStar's website. Just to show you guys two right here, we are hiring for a network engineer and also a full stack developer. If you're interested in applying for any of these roles, definitely check out our careers page and you'll find those open roles there. Or if you just want to find something else that might suit your skill sets, we have plenty of options available. And if you're interested in speaking at a future PyData DC meetup, definitely send me an email. My name is Cia Romero. I am the marketing coordinator here at MetroStar. So feel free to send me an email at marketing@metrostarsystems.com. And last but certainly not least, before we get started, we do want to give a thank you to our sponsors. We have PyData, MetroStar, and NumFOCUS. And thank you so much. So without further ado, I will pass it off to Xavier and we can go ahead and get started.
All right. Good evening, everybody. Pleasure to speak with you all. I want to give you all a talk on machine learning and biomedical microscopy. Let's see. All right. So this talk will be broken into 3 parts: an overview, discussion of methods, techniques, and applications. So the first part will go into my background as far as what is a full stack biotechnologist, followed by the value of biomedical microscopy, followed by motivation for deep learning uses in microscopy. So we'll then go into how classification, segmentation, artificial labeling, and image restoration can be very helpful for those doing microscopy in biomedical disciplines. Following, we then have applications in which we cover relevant cases for industry, basic science research boons, but also same for teacher engineering. And this will then fall into a collective discussion. I'm going to allow extra time for conversation, so this might be more brief than usual talks. All right, so for who I am Dr. Xavier-Lewis Palmer. My institutions are Old Dominion University and Eastern Virginia Medical School. In that, I've got a BA in biology. BA in philosophy, a BS in biology, a master's in cybersecurity, a master's in biotechnology, and then a PhD in engineering with a concentration in biomedical engineering. Some of my experiences include the National Institute of Standards and Technology, known as NIST, the ADA Foundation, and the Food and Drug Administration. My core issues are tissue engineering and cybersecurity, and I'm very passionate about the intersections that are between biology and security. Technologies that are between them, such as bioauthentication technologies, but also matters of protecting patient data and the wider bioeconomy are very important to me. So there's the question of what is a full stack biotechnologist? Well, that is a term that was defined and popularized by Stephen Lewis, and he gave a presentation at DEF CON 27 in 2019. He had a great talk called Cyber Biosecurity in the Full Stack Biotechnologist. A biotechnologist is an individual that can utilize software, hardware, and wetware. And by wetware, we're talking like biological and chem components, to use them all together to deliver quality biological products and outcomes. They can be essentially a one-man machine. Then in the right, you'll notice several skills that are crucial to those that are operating at the intersection, and you'll find that these individuals can can be very useful for not just for a solo operation, but also very well for they can work very well with helping entire companies. I recommend the talk, and the link is below. And also several of the links that I'll have throughout, I'll also place in the chat later on. So a full stack biotechnologist typically has a combination of self-taught skills, certification, and formal education. They're adept at working between fields and disciplines and understanding an entire pipeline. And they're also great for inserting themselves at any point in a pipeline to repair or enhance operations. Now, how these individuals come about, the backdrop for this is that the technology, such as recombinant DNA technologies or machine learning or just random, types of prototyping software, they've all become very much more accessible and cheaper. So it's easier for such individuals to make use of all of them. But specifically, the technology that I want to talk about more so with a lot of bio work is that of machine learning, specifically deep learning, which I've highlighted. So when it comes to biomedical microscopy, I'm talking about microscopy with a biomedical focus, especially towards like healthcare and basic science research. To assist, I'm presenting a paper that I recommend called A Comparative Study on the Use of Microscopy in Pharmacology and Cell Biology Research. And there they list a lot of data with regards to the types of microscopy that's used commonly within these areas, such as confocal, fluorescence, phase contrast, scanning electron microscopy, transmission electron microscopy, and many more. For anyone that's looking to get an idea of, okay, what might be attractive to work on as far as applying machine learning towards microscopy images, it's a good paper to start with. Now, all these types of microscopy, they allow us biologists and those adjacent to study cells and their components, especially their immediate environment and structures outside, especially the extracellular matrix, which you can think of as this, the outer structures around cells that allow cells to have more structure, that allow tissues to have more structure to facilitate nutrients transfer, but also communication, and so many more. They allow for our tissues to have this greater connectivity and functionality. But also, microscopy allows us to also better study the tissues that are important to us as well. Now, the use of microscopy in the biomedical sense allows us to make predictions, to render diagnoses, and also advance in basic science and engineering. So, I love microscopy, very fun. I will say that it can be very time consuming, but a way in which we can augment a lot of our workflows is with deep learning. There's another paper I'm going to recommend called Artificial Intelligence for Microscopy, What You Should Know, by Xavier et al. And I also have the link below there. They explore multiple things in this paper, but 4 that I want to go over with you are object detection and classification, image segmentation, artificial labeling, and image restoration and super resolution with respect to microscopy. So, but what are some motivations? Well, they can give you enhanced image quality and data sets, improved target acquisition, because you're going to be looking at a lot of things on a microscope, and the question is, well, how many things might you miss at eye level? But also, because of how time-consuming and tedious the work can be, there's also a lot of burnout. So you want to not burn out your health professionals, your scientists, your engineers, and so it's good to have people apply machine learning towards microscopy. As you move into a health discipline, it's really good for triage because you can have faster processing of patients as you're using these algorithms to go over various medical images. And you can also be able to catch details that you might otherwise miss. Numerous papers mention how a lot of these algorithms that are being trialed, they've been able to catch a lot of features that can be missed by people with years of experience in the field. Additionally, by the use of deep learning with microscopy, this may translate to less time billable and less institutional resources used, which can be a win-win for both the patient, but also the institution themselves. But as a researcher, as an engineer, as someone that's in between, but just loves to explore, I love how this can free up a lot of work, a lot of energy and time for a lot of other work that we can explore. And for me, the researcher, I would say I'm happy about all the extra space I have now to do more work and to ask deeper questions now that I can see more, that I can process more. In terms of, but for the institution, this is also win-win because you may find through this enhanced analysis and greater throughput that you may have, that you may find more cost-effective means, but also you might find more profitable parameters to hone in on. So everybody wins when it's done right, of course. All right, so classification is very helpful, especially when you want to identify and classify components rapidly, especially when you have a lot of samples to go through, but also when you're when there's there can be some ambiguity. Sometimes you can find that this will simplify your work very well. And there's 4 examples that I highlight that's also out of the same paper. They because in this review they mention various other researchers who have applied machine learning towards their projects. There is Ning et al. In 2005, the classification and segmentation of tissues and stages of C. elegans development. Pretty cool. Kraus et al. In 2016, high throughput classification and segmentation in yeast. Which is also very cool. And a lot of work with yeast is very cool, especially with synthetic biology. You have a lot of companies out there that are looking to use a lot of microbes as mini factories. You can think of like Ginkgo Bioworks and so many others where, something like this could be very useful. You have Dur et al. In 2016, phenotyping of over 40,000 drug-treated single-cell images. Now, I'll tell you all right now, I've done both but I've done manual, just manual identification of sets of images before, and I can tell you the use of machine learning algorithms has just cut a lot of my work down from like what might take weeks, like when I was doing some of this work as undergrad, and then down to like sometimes a few days or an afternoon, which is fantastic. To take 40,000 images and just run through them quickly, wonderful. Now, next, Parma and Parts 2017, high throughput classification of protein localization yeast, also very cool. It's good. Classification is very helpful. Next is segmentation, which there's I'm going to list some papers from the same list earlier. There's Keynes et al. In 2015 with colon gland segmentation, which the network outperforms handcrafted feature detection. It's much better, much simpler, and oh my gosh, way less way less stressful. Yeah, Van Valent et al. 2016, efficient cell segmentation and live cell imaging. This is also pretty cool too because there's a lot of advantages to live cell imaging over histology, and and in opposition to other means, because you're able to image the cells as they are, and it's cool to be able to segment, in real time. It's wonderful. Now, Naylor et al. 2018, application of UNet for nuclei segmentation in histopathology sections. So it's you'll find in a lot of microscopy applications, you're going to have to focus on what's happening in the nuclei, and that you can get segmentation of nuclei is nice too. In general, being able to, before I even say this point, I wanted to go over the point how there's a lot of things that we as scientists and research may miss by eye. And with a network that's properly trained to segment, you can find more features that you would otherwise miss. And as a result, you have more samples that you can study, and that can make for better stats and way better work. Now, probably my favorite out of these 4 is artificial labeling, because it allows you to like to bypass the limitations in the number of fluorescent labels applied to your sample, and allows you to also bypass the limitation of phototoxicity. For those who aren't in any wet labs or work with cells or in any similar capacity. In order to visualize certain parts of your cells or tissues, you'll have to stain them or apply some sort of labeling, whether and that could be in the form of antibody with a fluorescent signal attached. But ultimately, you're going to have a limitation with regards to the signals that you can read and or see. And also, there's a lot of complications figuring out the right chemistry between the different dyes or the different antibodies, etc. Depending on what you're doing, you're going to find that there are limitations that you can get past if you can digitally label, which you can do through machine learning, which is fantastic. But also, another point I bring up is the reduction of exposure of your biological samples to the environment. Some people might say, well, hey, don't you have your cells in a flask? Well, I mean, your your cells when they're in the incubator will be in a flask, but you got to think about the environment that your flask is going to be in. It's going to be in a environment that is mimicking physiological conditions. Your lab might be pretty cold, and as you can think of, like the many times you might take your flask out of the incubator into your cold lab or whatever situations are going on with your lab, there's different cycles of exposure indirectly that that can be a complicating factor that you might not know. But what if you are able to not have to apply, you're able to, you're able to keep your cells you're able to keep your flask in the incubator and just, artificially label from within. That can be a bonus. But also, if you don't have to take longer exposures, you can use much more simple imagery and then artificially label more basic images. That's less exposure that your samples have, like from moving from your lab, from your outer lab environment to your incubator and back and forth. But all of this is contextual depending on exactly what you're doing. So for some people, this may not apply, but I love the idea to artificially label. And also, there's a lot of work that can be moved in. There's a lot of work that can be made less intensive, which I appreciate. But also, in general, you can reduce your cost and time and analysis because you may not have to use as many fluorescent labels, or you may not have to use certain dyes, which is fantastic. Now, next is image restoration and super resolution. I enjoy that too. I'm going to bring an example from Bookos and Jug. They have a paper called Noise to Avoid, and I'm using an image in the left from their archive preprint. They eventually published, so I'm listing that paper below too. And in that, they show how that you can use machine learning as a means of reducing noise, taking out artifacts and specks, and cleaning up an image so that you can get a clearer picture, which is wonderful. Not much more needs to be said about this, but the general idea is that you can improve resolution and you can reduce the noise in images, which allows you to rehabilitate and use more images in your original set. Now, relevant case for industry. You can probably glean several of those out of basic science and research. But I'll say it again, for basic science and engineering research, you can find new and useful features in and from organisms and in greater amounts. In healthcare, this can help with sorting and identifying patient conditions. In publishing, you'll find that, hey, there's a replication problem, but also there's a lot of fraud that you may find in some health-based publications. You can use machine learning as a means of identifying where there's fraud in work. But as far as some alternate QA, QC processes, you can use machine learning as a means of verifying medical graft quality, looking at quality such as porosity, fiber diameter, etc. You can use machine learning as a means of eliminating samples in a run by looking for defects, which it can probably find better than normal human eye can. But also in biosecurity, of course, in a similar way, you can use that as a means of pathogen identification and quantification. In terms of a generative sense, you can use machine learning as a means of, especially through GANs, to make models or to make images for educational purposes. I can imagine a lot of teachers enjoying that. To the right, there's like various biologically relevant samples that I've developed in terms of like 3D culture, 2D culture, various like fibrous meshes, and more that indirectly can be the work can be enhanced by AI/ML depending on how you apply it. Um, once you image the once you image the biological samples, and so let's see. But yes, another work that I want to point out, which is amazing, is the cell atlas. Leica Microsystems has an interview with Professor Emma Lundberg, who's professor in cell biology proteomics at KTH Royal Institute of Technology in Sweden. She's also the director of the Cell Atlas, which is an integral part of the Swedish-based Human Protein Atlas program, which is an open source resource for human proteome exploration. They provide high-resolution images of expression in spatial temporal distribution of RNA and proteins in human cell lines. And one thing I'll pick out, and so I'm not going to give everything because I want y'all to read this because I believe her interview is great and helpful, is the importance of the streamlining of image processing and analysis. There's just so much more work you can do in a better way once you're using all these tools to help you sort through samples, image samples, and refine samples. AI is a boon for sure. So tissue engineering, that's largely the core of my my dissertation work. And for work that is related to that's either at PACE or within my dissertation, I will exclude that, just out of respect for my lab. But I can give I can give the general parts of it and how machine learning can be applied towards it. So my prior work in my dissertation and work leading up to it was in organoid development and the investigation of the microenvironmental control of cell fate. To slow that down is we're we're asking questions of how does the microenvironment change how a cell behaves? And as I mentioned earlier, in the microenvironment, it can be all the different fibers and compounds, and so providing structure, facilitating movement of vital, compounds to the cells and from, but also providing structure, the translation of so many physical forces. There's a lot. And yeah, that's very important for understanding not only regenerative medicine, but also for understanding, cancer biology. And for a lot of my work, it was understanding breast cancer. This work utilized plenty of programming, 3D bioprinting, and most importantly, multiple types of microscopy, including, but not limited to, phase, fluorescence, and confocal. The two images, like to the left, are from a interview that I had with Virginia Pilot, and I have a link above the top left picture that y'all I'd say feel free to read. It talks about the work that we were doing, and this is about 4 years back, but hey, it's very much relevant. And so you see me with my printer, and below you see one of the cell plates that with extracellular matrix that we printed into. To the right of that image, you'll see a time lapse of an organoid of a chimeric organoid structure that is developing. And so from 7 to 14 days. And this is from our publication, a 3D bioprinter platform for mechanistic analysis of tumoroids and chimeric memory organoids. Now, as you look at this image, you see a lot of crazy cool features. You'll see, okay, well, for this ring structure that we're creating, there's like all these sac bulbish-like structures, which are pretty cool. You see different colors. You see a lot of different geometries at play in this structure that we created. Machine learning can help not only with just better making sense of these images, and so, but also helping us to better segment and identify particular features that grow out of these. The citation from the paper is below. I strongly recommend y'all read that. It's great. And I will say that machine learning can not only help with just the improved resolution of the images and denoising, but indirectly through just the use of how we're examining these images, it can help towards the ultimate goal of improving organoid design. So for us to get up to where, well, I'll stop there, but all right. As far as additional newer works, yeah, there's a lot more works that are coming out, which are great. just by so many other scientists that are, especially biologists that are getting into applying machine learning to their workflows. There's the question of, well, do I just continue, just manually going through these images, and just work at a snail's pace, or do I jump in the fast lane? And I love it. So like one example is machine learning algorithms in neuroimaging and overview. So that review is pretty cool as far as like how these are being applied towards neuroimaging. There's another example is like YeastMate. They're applying a neural network to segment like mating and budding events, which is also helpful. I mentioned earlier how yeast is very important in labs and bioapplications. It's worth looking at. A bunch of DIC images and whatnot that are looked at. And then next is machine learning in scanning transmission electron microscopy. So you're seeing that for even more complex means of microscopy, more individuals are, more people are finding ways to apply machine learning workflows. Machine learning to their workflow, because yeah, we wanna do more, but we wanna do more efficiently, and I love it. So thanks for listening to my talk. Feel free to forward questions to Xavier-Polymer@proton.mail. The polymer comes from my earlier polymer research. I did a lot of polymer airbrushing, in which we created biomedical scaffolds, and so using airbrushes and biocompatible polymers. And my last name is Palmer, so you have the you can see how it translates. But yes, I will also place a link in the chat for everyone for links in the chat for a lot of the papers discussed earlier. Feel free to come through any questions, and thank you so much.